Hi everyone, welcome back to Biological Psychology. Today we will be talking about Chapter 2, which will focus on the neuron doctrine and the prototypical neuron. So first I will be discussing the neuron doctrine, but before we move on to this theory, I want you to know that there are two key types of cells in the nervous system. Of course, you know there are neurons. Neurons are very important because they can sense changes in the environment. They communicate with one another to help process neurochemicals from one cell to the next, and they command the body's responses to sensation. Glial cells, also known as glia, contribute to brain function by insulating, supporting, nourishing, and protecting neurons. In this course, we're going to learn about a lot of different types of glial cells and neurons, but I want you to think about glial cells and neurons like a chocolate chip cookie. Essentially, glial cells are like the cookie dough that hold the chocolate chips or neurons together. So neurons could not survive without the support of glial cells. And there are approximately 85 billion of each cell type in the brain. Neurons are extremely small. Most of the cells are within the range of 0.01 to 0.05 millimeters in diameter. So to put this in perspective, the tip of an unsharpened pencil is about 2 millimeters, and neurons are 40 to 200 times smaller than this. And the resolution of the unaided human eye without a microscope is about 100 micrometers. And I do want you to know the units of size in the metric system for this class. I could test you on any of these units. I do want you to know that the near limit of resolution for the light microscope is in micrometers, where the electron microscope is in nanometers. So we can't actually see a neuron with the naked eye because it's so small. So thus, we couldn't actually look at neurons and study cellular neuroscience until the light or compound microscope was created in the 17th century. The neuronal cell body is generally about 20 micrometers in diameter. The cell body contains the nucleus and the genetic material within the cell. The space in between neurons, however, is much smaller. It's actually only about 0.02 micrometers, which is 20 nanometers. So thus, we couldn't actually investigate the space between cells until the invention of the electron microscope that has a resolution in nanometers. First, we're going to talk about the prototypical neuron. So as you can see, you've probably seen a neuron that looks like this before. It has a cell body, which is also known as the soma. Soma and cell body are synonymous. So on your exam, you will have to label a neuron. And so if you labeled this the soma or cell body, both answers would be correct. The cell body usually gives rise to one single axon. Axons are these long branches that come off of the cell body, and they have to be very thick in diameter because this is what helps expedite cellular communication, which we'll talk about in a little bit. But essentially know that axons are super important for communication between cells. They can extend very far distances, up to a meter or more. So they act as wires that carry nerve impulses that are also known as action potentials, which we will cover in chapter 3, from one neuron to another. And think about it. Our bodies are very long. I'm a very tall person, even though you don't know that since this is an online class. I'm about 5'9.5", and we have to have axons that project from the brain all the way down to the nerves in our feet. So thus axons can be very long. Dendrites are rarely longer than 2 millimeters, and many dendrites actually extend from the cell body, and then they generally taper to a fine point. So they're very short, about 2 millimeters. So how do neuroscientists study the structure of neurons? This is through a technique called histology. Histology is the microscopic study of the structure of tissues. This image here is actually taken from the hippocampus of a mouse. The hippocampus is the key brain area for the neural mechanisms of learning and memory. And we could only study the structure of the hippocampus by taking extremely thin sections of the tissue that are usually within micrometers or nanometers in size to look at one single layer of cells. The cells are usually stained, as shown here, to look at the number of neurons or other types of cells like glia. Histology has really advanced science. We can view cells under the microscope due to this extremely thin section of tissue that's usually processed on something called a microtome. This is a video here of a cryostat microtome, and as you can see, the sections of brain tissue from this rat or mouse are extremely small. They need to be small because we only want to look at one single layer of cells. Here is another example of a microtome, which is used to take very thin sections of brain tissue. In a research setting, we can take brain slices from a rat or mouse to study the progression of disease pathologies or potentially look to see if therapeutic intervention reduce those disease pathologies. There are many other purposes for histology. For example, in medical settings, you may have histology conducted on maybe a tumor to look at the pathology, at the cytoarchitecture, and ensure that that the tumor is not cancerous or malignant. Histology has really helped us Before you can conduct histology, tissue needs to be fixed. This essentially means that tissue is hardened with chemicals like paraformaldehyde in order to do proper staining. Staining is essential because it helps us look at individual cells in our very thin sections of tissue. There are certain stains that can even stain for specific types of brain cells, like individual neurons. So, how do scientists stain neurons in glial cells? The first stain was invented in the 19th century by Franz Nissl. This was known as the Nissl stain. And Nissl bodies were actually clumps of nuclei of the cell structure. So this invention was important for two reasons. The Nissl stain helps us distinguish between glial cells and neurons, and it enables histologists to actually study cytoarchitecture, which is the arrangement of different types of brain cells in the tissue, such as the number of neurons in comparison to glial cells. Also, the study of cytoarchitecture led to the realization that the brain consists of many different specialized regions. And we now know today that each region does perform a different function. Another stain that was invented was called the Golgi stain. And the Golgi stain was very innovative. What he did was he soaked the brain tissue in a silver chromate solution, and it actually allowed him to see the entire neuron, not only the nuclei or the cell body, but the axons branching away from it. And so this revealed that the neuron was not just made up of this small, tiny cell body, as shown in the Nissl stain, but that the total structure of the neuron was actually elongated. He identified the two different parts of the neuron, including the cell body and the axon. So this stain was really influential in the field of neuroscience and psychology. This led to his theory. Golgi championed the view that neurites of different cells are fused together to form a continuous reticulum. So this would be similar to like arteries or veins in the circulatory system. However, this theory goes against the grain. In science, there is a theory that each cell works independently of one another and that individual cells are all their separate entities that work together. However, Golgi's reticular theory was an exception to the cell theory, and he thought that all neurons' axons were connected. Interestingly, after Golgi created the reticular theory, Ramon Cajal started using Golgi's stain to draw pictures of neurons. So Cajal was a Spanish neuroscientist and a very skilled histologist. And you can actually visit his museum in Spain, and he has beautiful drawings of neurons. He used Golgi's methods to work out many of the regions of the brain and their actual circuits. Interestingly, he theorized that neurons were actually not continuous, but they followed the normal cell theory accepted by biologists that neurons are separate entities that communicate by contact and not continuity. So, this idea that the cell theory also applied to neurons was known as the neuron doctrine, and this theory is now accepted today. Neurons are not continuous. They do, in fact, communicate by contact and not continuity. However, Cajal's hypothesis could not be proven until the invention of the electron microscope. So, over the next 50 years, we had to wait for the electron microscope to be created because if you remember earlier in my lecture, I talked about how the space in between neurons is extremely small. It's only about 20 nanometers. So the resolving power of the electron microscope proved that the neuron doctrine theory was correct and that neurons are individual cells that communicate with each other and they are not actually connected. So now this brings us to chapter 2, part 2, where we're going to discuss the structure of the prototypical neuron and neuronal morphology. So I know this class is virtual and sometimes you won't be coming in person, but I do want you to keep up with some of our pop quizzes we do in class. They're not graded, but I think if you go through this and you can try to answer all of these questions, it will help you with your studying and keep on top of our current class material. So this is a great way to test yourself and see if you're retaining the information from our previous lecture. Now I'm going to talk about for your first exam, you are going to need to label the prototypical neuron. So as you can see here, this is the soma or cell body. These two phrases can be used interchangeably. The cell body contains the nucleus, and inside of the nucleus is the cell's genetic material. We'll talk about the importance of this throughout this lecture. And then there are dendrites that come off of the soma or cell body. And we're also going to talk about the importance of the little dendritic spines that are on dendrites and the role they play in cognition. You'll also need to label the axon proper, which is this long strand here. And the axon needs to be very thick in diameter because it is insulated by something called the myelin sheath. Myelin helps to speed up synaptic transmission and communication between neurons. We'll talk about the formation of myelin and the certain types of cells, such as Schwann cells, that help to create it. At the end of the axon, this is called the axon terminal or terminal bouton, and we'll talk about what occurs at the axon terminal later in this lecture. So the cell body is really small. It's typically about 20 micrometers in diameter. So you wouldn't be able to see it with the naked eye, and that's why the light or compound microscope was so beneficial for our field, so that we could actually look at individual neurons. The inside of the cell body is filled with cytosol, which is water filled with ions like salt and potassium. And we're going to talk about the importance of these ions in setting nerve impulses later on. Inside of the cytosol, the organelles are these membrane enclosed structures that are floating in the soma. And there are several different organelles we're going to discuss, including the nucleus, which holds DNA or the genetic material, the rough endoplasmic reticulum, which is a key site for protein synthesis, the smooth endoplasmic reticulum, the Golgi apparatus, and mitochondria. So here's the cell body of a neuron. And as you can see, the nucleus is the very center of the neuron. The nucleus is protected by an envelope outside of it called the nuclear envelope. Within the nuclear envelope are these small pores known as nuclear pores. This allows any type of information that needs to go outside of the nucleus into the rest of the cell in and out. Right outside of the nucleus is the rough endoplasmic reticulum, shown here in purple. And in the rough endoplasmic reticulum, it's known as rough ER because small ribosomes come and attach to the rough ER here. Ribosomes are key sites for protein synthesis, which occurs right outside of the nucleus. We'll also talk about other organelles like the Golgi apparatus that help direct proteins where they need to go, and the mitochondria. As we all know, it is the powerhouse of the cell, but this is because it creates adenosine triphosphate following the Krebs cycle. Adenosine triphosphate, or ATP, is the cell's key source of energy. As I stated previously, the nucleus houses our genetic information, which is a genetic code called DNA. DNA is a double helix of nucleic acids that are stranded together to assemble something called genes. The reading of these patterns of nucleic acids in DNA is known as gene expression. For the purpose of biological psychology, I just want you to think of DNA like a template with specialized codes that are made up of nucleic acids. These nucleic acids here form base pairs in the double helix structure of DNA to code for genes that will later be translated to protein. DNA contains the entire genetic code, or genome, for the body. But why is gene expression so important? Well, gene expression codes for proteins that are vital to the body and brain, and we couldn't function without protein synthesis occurring. The assembly of proteins occurs in free-floating ribosomes or on ribosomes that attach to the rough endoplasmic reticulum. Remember, the rough ER sits right outside of the nucleus of the cell. Ribosomes can also form into chains together called polyribosomes. So how does protein synthesis occur? This is very important because DNA can never leave the nucleus. So how do genetic messages reach the rough endoplasmic reticulum outside of the nucleus? First, DNA needs to be transcribed into messenger RNA, as shown here. So messenger RNA is a single strand of nucleic acids that are created from the code and the sequence of nucleic acids in DNA. This represents our gene information. So how is messenger RNA assembled? Here is the central dogma of our biology. We've discussed DNA and how it never leaves the nucleus. Genetic information is computed from DNA to messenger RNA and then to protein. So protein is essentially the way that genes are expressed. First, we're going to talk about how DNA is transcribed to a single strand of messenger RNA. During the transcription process, first, RNA molecules need to be synthesized, and this is done with the initiation of RNA polymerase. RNA polymerase is an enzyme. Enzymes help to catalyze reactions. So essentially, this enzyme, RNA polymerase, comes and binds to the promoter region of DNA. The promoter region is the start of a specific sequence of mRNA, and this initiates the transcription process. Once transcription is occurring, there will be the removal of certain segments within DNA. For example, exons are the segments that are going to end up coding for protein, so they remain in the sequence. However, introns are spliced out of the mRNA because they do not code for protein. Additionally, I want you to remember that transcription initiates at the promoter region of the gene. When RNA polymerase comes and binds to this area, it is known as a transcription factor because it will initiate the transcription process, which essentially just means that introns will be spliced out of DNA so that exons can remain. The exons are just simple strands of nucleic acids that will be coding for a particular gene in the mRNA. The initial RNA must be sliced to remove the introns because they do not code for protein. Once this is done, mRNA can be exported from the nucleus, meaning that it's going to leave the nucleus through these nuclear pores to be able to go to free-floating ribosomes or to the rough endoplasmic reticulum for protein synthesis to occur. Essentially, a big takeaway from this lecture, I want you to know that genes, mRNA, carry codes to synthesize proteins that are made up of amino acids. So the nucleic acids will be translated to amino acids. How does this step occur? This is through translation. So again, we've already discussed how DNA is transcribed into single-stranded messenger RNA because this allows it to leave the nucleus. Here's the nucleus of the cell up here, and mRNA can leave through nuclear pores. Now, after it leaves the nucleus, it's going to either float over to a ribosome or the rough endoplasmic reticulum where ribosomes have bind to initiate protein synthesis. Amino acids then form a chain and proteins are assembled. This occurs at the ribosome site. I myself am a visual learner, and I like to provide you with the tools to visualize all of the biological processes we're going to be describing in this class. So remember, we have 23 base pairs of chromosomes. Chromosomes contain all of our genetic material, or DNA. DNA is very long, so it needs to be tightly bound by histones so it can become more compact, so it can fit in the chromosome. Remember that DNA has certain segments that contain strands of base pairs of nucleic acids that code for particular genes. When RNA polymerase, the enzyme, comes and binds to DNA, remember this occurs at the promoter region to initiate transcription, the double helix DNA is transcribed into single-stranded messenger RNA. Essentially, messenger RNA contains a pattern or sequence of nucleic acids that are going to later code for proteins. Messenger RNA is created in the nucleus, but it has the ability to leave the nucleus through the nuclear pore. Once messenger RNA is completely assembled, it leaves the nuclear pore and goes outside of the nucleus. So it is either going to make its way to a free-floating ribosome or to the rough endoplasmic reticulum where ribosomes have already attached. And this is the site of protein synthesis. I don't need you to understand all of the different enzymes or types of RNA that are playing a key role here, but what I do want you to know is that the messenger RNA has nucleic acids that are going to help create the pattern necessary to translate those nucleic acids to amino acids, because proteins are simply made up of amino acids strung together. Amino acids, in the order or sequence they are created in, provide the code for the type of protein they are creating. As you can see, all of these tiny purple amino acids are coming together to form a protein. There are several different types of protein structures we're going to talk about in this class. We're also going to talk about how the misfolding of protein can be related to disease pathology. Also, I want you to know that specific genes code for very particular proteins that are needed in the brain. We know that the human genome, which is the entire length of the DNA in our chromosomes, is composed of over 25,000 different genes. And that's why it's so essential that we have histones that can tightly compact our DNA into very small areas to fit inside the chromosome. So what happens if genes are missing or duplicated? This is called the gene copy number variation. This is associated with several different psychiatric disorders, such as autism and schizophrenia. So this occurs if a gene is missing or if it's simply duplicated. What happens if a mutation occurs in DNA? This is a new research area that is really growing. We're finding that several different disorders and neurodegenerative diseases are linked to genetic mutation. So think of it like this. What would happen if a code was missing for a single protein that should be transcribed or translated? This is called a single nucleotide polymorphism, or SNP. Sometimes a SNP does not actually affect a person, and you won't see any physiological, phenotypical, or behavioral changes. However, there are several that do present phenotypical differences, and this is induced by a single letter change in a strand of DNA. An example of a SNP could be Fragile X syndrome. This is a disorder that manifests itself as intellectual disability and autism from disruption of one single gene. And as you can see, in addition to inducing intellectual disability, it also presents phenotypical traits and changes in a facial structure. And this is caused by a single letter change. However, most SNPs do not affect a person, and you will not actually see physiological or behavioral changes from the single letter change. Next, we move on to the Golgi apparatus. So remember, newly synthesized protein is most likely made in the rough endoplasmic reticulum where ribosomes have attached. Now, newly synthesized protein goes to the Golgi apparatus, which functions like the post office service of the cell. And it sends the proteins where they need to go. Also, I want you to know that the mitochondria is very important for creating cellular energy. Through the Krebs cycle, mitochondria produces an energy source called adenosine triphosphate, or ATP. I don't expect you to understand how the Krebs cycle works for the context of this class, but I do want you to know that the mitochondria produces the cellular energy. This is going to be really important in chapter 3 when we talk about the energy needed in the sodium-potassium pump, because it keeps the membrane at resting potential, but we will get to that in chapter 3. What happens when protein assembly is disrupted? One of the most common pathologies of Alzheimer's disease is due to a misfolded protein called amyloid beta. Misfolded amyloid beta protein induces extracellular amyloid beta plaques. Here is an image of neurons that are trying to communicate, but their communication is disrupted because this extracellular clump of protein is in the way of their axons. The two hallmark pathologies that Dr. Alzheimer discovered in the early 1900s include amyloid beta plaques, as shown here, and hyperphosphorylated neurofibrillary tau tangles, shown here in the purple circle. So interestingly, these images were taken from a post-mortem brain, meaning that this patient did die of Alzheimer's disease. So these are real plaques and tangles. Amyloid beta is a peptide that is originally produced as a monomer, meaning that it's a single protein. However, in a pathogenic state, there's misfolding of protein, meaning that amyloid beta can clump together to form fibrils or plaques. These plaques form extracellularly, meaning they are created outside of the neuron and thus disrupt communication between cells. Additionally, tau is an important protein we're going to talk more about on the next slide, and it's hyperphosphorylated, meaning an extra phosphate group is added to the protein, which essentially creates tangles inside of neurons. So to better understand how the protein tau is involved in Alzheimer's disease pathologies, I want to talk about the role that microtubules play in neurons. Microtubules are very important for creating the cytoskeleton, or basically structure, of the neuron. Microtubules help a neuron maintain its shape and aid in axonal transport of important proteins or chemicals. So essentially, microtubules are made up of tiny proteins called tubulin. And this is absolutely necessary to keep the shape of the neuron and to help with the transport, again, of important proteins for neuronal communication. So there's one class of these proteins called microtubule associated proteins, or MAPs. And MAPs help to anchor the microtubules to one another and to other parts of the cell. Pathological changes in one of the axonal maps, called tau, has been implicated in Alzheimer's disease. Tau is normally a protein that helps with the cytoskeleton structure and exoplasmic transport of information across neurons. Usually it functions as a bridge, as shown here in this left side of the image. This is a healthy neuron. So microtubules, again made up of tubulin proteins like MAPs, ensure that all of these microtubules run parallel to one another and help the axon maintain its shape. In Alzheimer's disease, tau proteins can actually detach from the microtubules and accumulate in the soma or the cytosol. This disruption of the cytoskeleton causes axons to wither. They lose their structural shape and function, and they're no longer able to communicate with other cells as this affects neurotransmission. There are specialized proteins we're going to talk about later on in today's lecture that help aid the transport of important neurotransmitters or proteins through synaptic vesicles across the axon, and without the correct shape, this cannot occur. This is known as hyperphosphorylated neurofibrillary tau tangles, and this is the other key pathology of Alzheimer's disease. As you can see, there are signs of AD pathology all over this brain. The small orange dots demonstrate different pathologies. However, the two hardest hit areas for Alzheimer's disease include the medial temporal lobe, which is home to the hippocampus. The hippocampus is a key brain region for learning and memory processes. Additionally, the prefrontal cortex develops pathologies as well. And so, thus, this is why Alzheimer's disease patients do struggle with memory loss and cognitive impairment, because many of the brain's memory systems are compromised by these misfolded proteins that develop into AD pathologies. Now we're going to move on to the classification of neurons. First, let's quickly review neuronal genes and genetic variation. These are all key terms you should know for your first exam. I want you to know that we have 23 pairs of chromosomes, and our long strands of DNA need to fit inside of chromosomes. Chromatin are tightly bound genetic material that make up our chromosomes. Our DNA strands are so long that they need to be coiled up, and histones play an important role in helping to keep these chromatin tightly compact. Remember that DNA contains introns and exons. When DNA is transcribed into messenger RNA, introns do not code for protein, and thus they need to be spliced. There is an alternative type of splicing, literally called alternative splicing, in which introns remain in messenger RNA, but for the purpose of this class, we're not going to get into that. What I want you to know is that during the splicing process during transcription, introns are removed and exons stay within the genetic code. So remember, when messenger RNA is transcribed, all of the exons remain, and these are segments of the genetic code made up of nucleic acids that will eventually code for protein. So how do neuroscientists study specific alleles and gene mutations associated with neurodegenerative diseases? An important breakthrough for studying disease was through genetic engineering. This is a huge breakthrough for the field of neuroscience and psychology because we can change organisms by creating animal models. An animal model can have a gene inserted into its genome so we can study disease progression and potential therapies for it. So we can actually insert a particular gene from humans into the mouse genome, or we can even delete or knock out a gene to better understand how that one particular gene works and affects the body. This technology is used primarily in mice because they rapidly produce and they have a very similar central nervous system to us. That's why they're called a model organism. There are several different mouse models that have been designed, and we're going to talk about two in particular. The first, they're called knockout mice. This is when one gene is removed from the mouse genome to study the progression of a disease. For example, if we know that there's a specific gene that is correlated with a disease in humans, we can completely remove it from a mouse genome to see how it affects the animal and how it plays an important role in that model system. Transgenic mice, on the other hand, have genes that are actually introduced into their genome and they're overexpressed. These are called transgenes. So essentially, transgenes are actually taken from human genes and inserted into the mouse genome. An example of this is the 5XFAD mouse. I've worked with this mouse model before, and this stands for a familial Alzheimer's disease mouse model. There are two different types of Alzheimer's disease. This includes sporadic Alzheimer's and familial Alzheimer's. Familial Alzheimer's disease is associated with several different types of gene mutations, including mutations on the gene amyloid precursor protein and presenilin 1. Both of these genes are involved in the cleavage process and formation of the protein amyloid beta. If you remember a few slides back, we talked about how amyloid beta can be misfolded and clumped together to form extracellular plaques. So these genes, mutations in these genes in humans have been associated with increased risk of familial Alzheimer's disease. However, mice do not develop Alzheimer's disease, so we can create a model organism by actually taking these mutations from humans and then overexpressing them or simply amplifying them in the mouse. This particular mouse model actually develops Alzheimer's disease pathologies. It will develop amyloid beta plaques and cognitive impairment and memory loss, similarly to a human. So why is this helpful? This is great because we can use different drugs or therapies in the lab to understand how a therapy could be more therapeutic in slowing down pathologies or preventing further progression of them. Additionally, we can utilize these models to find target treatments and therapies that could potentially be moved on to clinical settings to help our loved ones and other patients who are struggling with Alzheimer's disease. So now that we've learned about all of these molecular processes that occur inside of the neuron, we can understand how neurons communicate with one another. So now I want to talk about axons a little bit more. So we've already talked about how axons are these long branches that project from the cell body or the soma, and how they play a really important role in transmitting information from cell to cell. They can cover great distances throughout the body because they can communicate with cells from all the way from the brain down to other peripheral systems. So we're first going to look at the axon hillock. This is the area that sits right underneath the soma. This is where the axon begins. The axon proper is this long branch that stems down. It's important to note that there are no rough endoplasmic reticulum in the axon, and there are very few free-floating ribosomes, so thus protein synthesis does not necessarily occur in the axon. It primarily occurs in the soma or cell body. So the protein composition of the axonal membrane is quite different from that of the soma. Axon collaterals are branches that actually stem off of the axon itself. And there are two different types of collaterals I want you to know about. First, collaterals can synapse onto either the neuron that gave rise to it, or they can synapse on neurons around it. Occasionally, an axon collateral will actually synapse back onto the soma or cell body that gave rise to the axon that it is attached to. This is called a recurrent collateral. An axon collateral that simply communicates with neighboring cells is just called an axon collateral. So there are synapses that can occur between the neighboring cells. Know what axon collaterals and recurrent collaterals are. The axon terminal is the very end of the axon. So here's the axon proper, and then at the end of the cell, the axon collateral plays a really important role because this is where neurons can communicate. Electrical signals, which we will learn about in our next chapter, are called nerve impulses or action potentials, and they can move at a quicker speed depending on the axon's diameter. This is what helps cells communicate. So the thicker the axon is, which means that it has more myelin sheath, which don't worry, we're going to talk about how myelin is created later on. But the myelin sheath that insulates this can increase the nerve impulses, which basically ensures that information is transmitted quicker. So all axons have the beginning, which we've already talked about. The axon hillock sits underneath the soma or cell body. The axon proper is this branch, and then the axon terminal is also known as the terminal bouton. So this is a key site for contact between cells. If you remember, there's a very small space in between neurons. It's only about 20 nanometers. Whenever we're talking about the synapse is where the cells pass information to each other. This comes from the Greek meaning to fasten together. We'll often refer to something as a presynaptic cell and a postsynaptic cell. At the synapse, a presynaptic cell is always trying to transduce information to the postsynaptic cell. So the synapse is where information is transferred from one neuron to another. And this is a really key point I want you to know. Here is a model synapse. Here is the presynaptic cell, and it needs to send information down here to the postsynaptic neuron. This entire area is the synapse. So here's the axon proper, and then we know this is the axon terminal. At the axon terminal, there are these specialized proteins called vesicles. This is the synaptic vesicle, and we can pretend that it's carrying an important neurotransmitter such as serotonin. Synaptic vesicles have already traveled down the axon proper and need to come down to the bottom of the cell. When they enter the axon terminal in the presynaptic cell, they are going to leave the neuron of the presynaptic neuron and enter the synaptic cleft. And this is that very tiny space in between the presynaptic neuron and postsynaptic neuron that's only about 20 nanometers in size. Then neurotransmitters can bind to receptors on the postsynaptic neuron and be let through into that cell. So this helps with synaptic transmission. In this example too, this is showing calcium entering through the presynaptic cell to the postsynaptic cell. This is a process called exocytosis, and we're going to talk more about exocytosis in our later chapters. All I want you to know from this is that this is an entire area of the synapse and that the presynaptic neuron needs to help facilitate synaptic vesicles carrying important neurotransmitters or other ions down to the axon terminal, where it will enter the synaptic cleft and eventually get to the postsynaptic neuron. There are several different types of synapses in the central nervous system, and so we're going to cover a couple of them today. First, if one neuron has an axon that synapses onto a dendrite of a neighboring neuron, this is called an axodendritic synapse. This is the most common synapse. Synapses are usually distinguished with where they're occurring throughout the body. In image B here, if the postsynaptic membrane is on the cell body, meaning that the axon is synapsing onto the cell body of a neighboring neuron, this is known as axosomatic. This inhibits the activity of other neurons. In some cases, the postsynaptic membrane actually will synapse onto another axon, meaning that one axon will synapse onto another axon of a neighboring cell. And this is known as axoaxonic. This inhibits the activity of other neurons. And we're going to talk about the difference between excitation and inhibition. Both are important in the brain. We never want to overexcite the brain, and we don't want to completely inhibit it. So specialized neurons can play a role in either excitation or inhibition. There's also a very unique type of synapse that usually only occurs in specialized neurons. And these are called dendrodendritic synapses. They're usually found in the olfactory bulb. The olfactory bulb is in charge of olfaction, such as registering smells. And this is a key site for neurogenesis in the brain. Ultimately, dendrodendritic synapses usually occur in these specialized cells. We're also going to talk more about exoplasmic transport. So if you remember earlier in the lecture, I talked about how microtubules play a really important role in the axon's shape and cellular structure. Well, on these microtubules, which if you remember are made up of tubulin protein, there are specialized proteins that actually work to carry and transport synaptic vesicles across the axon. There are two different types of transport mechanisms, and they're fueled by adenosine triphosphate, which, if you remember, this is the cellular energy created by mitochondria. The first is called anterograde transport, and this is done so with the protein called kinesin, shown here. Kinesin simply assists in the transport of synaptic vesicles. So let's give an example here. Perhaps the synaptic vesicle is carrying the neurotransmitter dopamine. Kinesin will help transport dopamine from the soma of the neuron to the axon terminal. This is called anterograde transport. Conversely, retrograde transport means that information or neurotransmitters are being transported from the axon terminal back up to the soma or cell body. And so in this case, for retrograde transport, there's a protein called dynin that helps carry proteins retrospectively. Retrograde transport has been thoroughly studied in research. So how can we look at this? Researchers discovered that if you inject something called enzyme horseradish peroxidase, HRP, it can actually be taken up and absorbed by neurons. So for example, if you deposited HRP into the brain, there are going to be neurons that are going to take up this fluorescent tag essentially via retrograde transport. So this is a way that we can see how cells absorb a substance and how it's transported throughout the axon. So some viruses actually utilize retrograde transport to infect neurons. For example, herpes virus, which produces cold sores, will actually enter the axon terminals in the lips and mouth and then transport them back to the trigeminal nerve. Rabies also travels via this system. So remember, retrograde transport is when herpes virus would enter the axon terminal and get back to the soma or cell body through the help of dynin protein. Shingles can also be transported this way, and they are transported back to the dorsal root ganglia. So if you remember, the dorsal root ganglia are cell bodies in the dorsal root nerve of the spinal cord. The dorsal root helps us understand sensations that we may feel in peripheral areas of the body. So shingles can be transported via retrograde transport and live in the dorsal root ganglia for decades. And so because they can live dormant for so long, that's why some people may be infected but won't present symptoms until they're older. Additionally, I want you to know that dendrites of a single neuron can branch together. So it's tiny little dendritic branches can branch together to form dendritic trees. And so we are just now discovering the importance of dendrites and dendritic spines. Particularly, we have found that dendritic spines are important for neural communication and are often assisting with receiving input from other neurons. So in addition to axon collaterals, which are going to be branching off to neighboring cells, dendritic branches and dendritic spines can also assist in this communication process. So dendrites are essentially like the antenna of the neuron. They're covered with thousands of synapses. And interestingly, they talk about this in your textbook, that there are actually polyribosomes that have been found inside of the dendritic spines. If you remember, polyribosomes are ribosomes that cluster together, and this can be a site of protein synthesis. So this is really interesting because we originally only thought that protein synthesis largely occurred in the soma or cell body, but it can also occur in the dendritic spines. And so there are receptors that exist on the dendritic membrane and other areas of the neuron that can help detect neurotransmitters in the synaptic cleft. Additionally, we have found that there are several different types of dendritic spines. I don't expect you to memorize all the morphology of all these different types of spines, but I just wanted you to see that there is a large variety. So why is this important? Well, researchers have found that dendritic spine morphology is directly related to cognitive function. So on the left, this is a dendritic spine from a cognitively normal infant. As you can see, there are many dendritic spines and they're very full and abundant. In the image to the right, this is dendritic spines from an intellectually disabled infant. So researchers found that infants that were presenting these changes, such as intellectual disability, had fewer and thinner spines. There are several pre-existing medical conditions that can actually affect dendritic spines. For example, if a baby is born with fetal alcohol syndrome, in which it was exposed to alcohol throughout gestation and lactation, this can reduce spine count and spine density. However, I do want to note that researchers are striving to find new therapies to actually reverse these effects and increase dendritic spines. So there have been therapies that have been utilized primarily through nutrition that can increase the growth of dendritic spines in intellectually disabled infants. I've also looked at dendritic spines in my doctoral research in the context of Alzheimer's disease. So, as I previously stated, I worked with the 5XFAD mouse, which is a mouse model for familial Alzheimer's disease, meaning that it has several gene mutations that were taken from mutated Alzheimer's disease genes in the human genome and inserted into the mouse genome. So essentially, you can think of these mice as the perfect model for familial AD because they develop Alzheimer's disease pathologies. Interestingly, I did a study where I compared the dendritic spines of a healthy animal. So this is a healthy animal model that did not have Alzheimer's disease transgenes inserted into its genome compared to an animal with the Alzheimer's disease transgenes. As you can see, the healthy animal had thicker and fuller dendritic spines. However, the animal that was developing Alzheimer's disease pathologies had fewer spines. Thus, we found that these animals not only had lower amounts of dendritic spines, but they also presented signs of cognitive impairment and learning disability. Thus, we have been able to correlate dendritic spine density not only in humans, but also in mice to cognitive impairment. Neurons can be classified in several different ways, and we're going to talk about all of those today. First, I want you to remember that there's two broad classes of cells. These are commonly found in the cerebral cortex. This is the structure that lies under the surface of the cerebrum. And if you remember earlier, I talked about how Galen studied the cerebrum in the sheep brain. So there are two broad classes. First, there are stellate cells, and they're star-shaped because this is the shape of their cell body. And pyramidal cells have a pyramid-shaped cell body. So these are two key broad classes of neurons. Neurons can also be classified according to how many dendritic spines they have and the length of their axons. Those neurons that are spiny are called spiny neurons, and those who do not have dendritic spines are called aspinous. So let's learn more about the actual dendritic classification schemes. In the cerebral cortex, there are pyramidal cells, meaning they have this pyramid-shaped soma. And in the case of the layer of cells in the cerebral cortex here, all of the pyramidal cells are spiny, meaning they have dendritic spines. Conversely, stellate cells can actually be spiny or aspinous. There are several different classifications of neurons. So these are the key morphologies I want you to know for the context of this class. So unipolar cells usually just have one long axon projecting from the cell body or soma. There are also pseudo-unipolar cells that have two different axons. These are commonly found in the dorsal root ganglia, and we'll talk more about that when we get to the spinal cord. Also, bipolar cells have two long projections of axons. There are also three types of multipolar cells. This cell is usually found in the spine. This is a spine motor neuron, and it kind of looks like your prototypical neuron. Hippocampal pyramidal cells, as shown here, have a pyramid-shaped soma, and they have plentiful dendritic spines. As we know, dendritic spines play an important role in cognition, and because the hippocampus is a key brain region for the neural mechanisms of learning and memory, we can understand why these pyramidal cells would have plentiful dendritic spines. The third one is the Purkinje cell of the cerebellum. The Purkinje neuron is actually one of my favorites just because it's so beautiful. If you cut down the cerebellum, you can see that there are all of these beautiful dendritic branches. This has been referred to as the arbor vitae or the tree of life because all of these dendritic branches are so intricate in the cerebellum. There are also different classification schemes we're going to talk about. So we've already discussed that neurons can be classified by their soma shape, meaning their cell body. For example, we've discussed stellate neurons that have a star-shaped body and pyramidal neurons that have a pyramid-shaped body. They can be classified on whether they're spiny or aspinous, meaning if they have dendritic spines, but they can also be classified by their axon length. And this is referred to as Golgi type 1 and type 2 neurons. If you remember, we talked about the Golgi stain and how it was the first stain that allowed us to look at the entire neuron. The silver chromate solution allowed us to not only see the cell body, but it also allowed us to look at the full axon. We know that there are other classifications like the functional type, if it's an inhibitory or excitatory neuron. Some neurons are defined by the exact neurotransmitter that they synthesize and process, which we'll talk about later on in this class, and sometimes they're classified on where they're located in the nervous system. For example, Purkinje neurons are usually only in the cerebellum of the brain. So as I said, Golgi type 1 neurons, Golgi type 1 and 2 neurons refer to the length of the axon. And so in these two pictures, first we're going to look at the pyramidal neuron. Pyramidal neurons are commonly found in the cerebral cortex and the hippocampus, and they usually have very long axons that can extend to other parts of the brain. It makes sense that neurons in the hippocampus would need to extend to the cortex, as this is a key pathway for processing memory. So, pyramidal cells usually have long axons that are known as Golgi type 1 neurons. And conversely, Golgi type 2 neurons are known as local circuit neurons because they have very tiny axons, as shown here in the stellate cell, that usually don't extend to a different brain region. So for example, this stellate neuron here would, if it was in the cerebral cortex, it wouldn't extend to other brain regions, only because it has shorter axons. This is known as a Golgi type 2 neuron. So spiny neurons are more likely to be excitatory, and if they have less spines, or if they don't have any spines at all, they're more likely to be inhibitory. Again, we've already talked about how neurons can be classified by their length, and you need to know what Golgi type 1 versus type 2 neurons are. Remember that type 1 axons can extend to several different areas of the brain, so they can synapse onto something called distal dendrites, where type 2 axons are shorter, such as on the stellate cell, and they don't travel far, thus they synapse onto proximal dendrites. And so this is very important for understanding the projections of neurons throughout the brain. It's also really important to understand how neurons connect, because neurons are taking in different sensations throughout the body, sending them back to the brain, and then motor neurons are going to play an important role in commanding our muscles that require movement after feeling these sensations. Neurons that are responsible for the delivery of sensations to the brain from the rest of the body, such as the skin or the retina of the eye, are called primary sensory neurons. Other neurons that have axons that form synapses with the muscles and command movements following sensations are called motor neurons. And so a majority of motor neurons are located in the motor cortex, in the brainstem, and in the spinal cord. And if you remember, it's the ventral roots in the spinal cord that are helping command muscle movements. Motor neurons actually release a neurotransmitter known as acetylcholine. And so acetylcholine was actually the first neurotransmitter ever discovered, and cells that release acetylcholine are known as cholinergic cells, meaning that they express the genes to enable the use of the neurotransmitter acetylcholine. This is really important at the neuromuscular junction, and when we get to neurotransmitter systems, we'll talk more about acetylcholine. But essentially what I want you to take away from this slide is that primary sensory neurons are the neurons that take in sensation from the body. Maybe if you accidentally step on something sharp, primary sensory neurons are going to send that information up to the brain via the dorsal root, and then the ventral root in the spinal cord will induce motor movement via motor neurons. So most neurons in the nervous system form connections with other neurons. In this scheme, they're called interneurons. This isn't always the case. Sometimes neurons can form connections with other brain cells, but most of the time they're forming connections with other types of neurons. So now we're going to talk about my other favorite brain cell, which is glial cells. Glial cells are super important because they completely provide the structure in the brain to support neurons, to protect them. They provide literal structural support because if you remember, I refer to glial cells like the chocolate chip cookie dough, whereas the neurons are like the chocolate chips within the cookie dough. They help transfer important nutrients. They can actually remove excess neurotransmitters in the synaptic cleft, and we'll talk about why that's important later on in this class. They help to form the blood-brain barrier. They help repair injuries in the brain. They provide the myelin sheath. We're going to talk about two types of glial cells in both the central nervous system and the peripheral nervous system that help create the myelin. And if you remember, myelin is what insulates axons and speeds up the nerve impulses that are occurring. They can also remove dead neurons and debris in a process known as phagocytosis. They can initiate the immune response in the brain. This might be interesting to some of you because it was not always well known that the brain had immune function. Most of science up until the last few decades primarily believed that the brain was an immune privileged organ, but that's not the case. And we'll talk about cells that function as immune cells in the brain. And then glial cells also play a really important role in neural migration during development. So here are the cells I want you to know. We have astrocytes, oligodendrocytes, and microglia. So first, let's talk about the role. So astrocytes play a really important role in maintaining the blood-brain barrier. They surround the capillaries of the blood-brain barrier with their little tiny end feet. They can help tell endothelial cells to tighten. Endothelial cells must create tight junctions to create a healthy blood-brain barrier. The blood-brain barrier helps to prevent toxins from entering the brain. So even though these end feet don't necessarily make up the blood-brain barrier, they work with endothelial cells to ensure that they are tight and that there are no loose gap junctions. This prevents anything harmful to get to the brain. So they support neurons, they can help repair damage, they regulate neuronal communication, and they maintain the blood-brain barrier with their glial end feet. This is not to be mistaken with endothelial cells. I want to make this very clear that endothelial cells make up the blood-brain barrier, but the astrocytic end feet do help to tighten gap junctions to ensure that nothing bad gets to the brain. This ultimately protects the blood-brain barrier. Astrocytes have a stellate body. So if you remember, that means they're star-shaped. We've already talked about their end feet and how they play a role in the blood-brain barrier, but their end feet also function instead of synapses. They help with chemical buffering and structural support for neurons. They also help remove neurotransmitters from the synaptic cleft. This is called glial cell uptake. Earlier, we talked about how presynaptic neurons carry synaptic vesicles of neurotransmitters down to the synaptic cleft in between the cells. This is how neurons communicate. Here in orange, we have a presynaptic neuron, and down here we have a postsynaptic neuron. And as you can see, these synaptic vesicles are carried down the axon to the axon terminal where neurotransmitters can be released. Another specialized postsynaptic neuron can take up this particular neurotransmitter. In this case, it's glutamate. As you can see, the astrocyte is providing structural support to the neuron as it sits tightly in between the cells. It's providing the support, but it also can play a role in chemical buffering. For example, glutamate is excitatory. If you have too much glutamate in the brain, this could lead to a seizure. So, astrocytes can also work around neurons and take up any excess glutamate that's left in the synaptic cleft. Astrocytes can also suck up extra potassium for chemical buffering to prevent overexcitation. And don't worry about this quite yet because we'll talk about the importance of excitation in future lectures. So astrocytes primarily fill the space in between neurons, so that's why I've referred to them as the chocolate chip cookie dough, and neurons are the chocolate chips. So essentially, they're not only providing structural support, but they're regulating the chemical content in that extracellular space, like we just talked about with glutamate in the previous slide. Astrocytes do regulate the concentration of both potassium ions and glutamate in the extracellular fluid. And so these are two that I particularly want you to know. Astrocytes also help out the blood-brain barrier. The blood-brain barrier itself is formed by endothelial cells. However, the astrocytic end feet will help provide structural support. So here is our endothelial cell in this blue color, and these purple cells here are the astrocytic end feet. As you can see, the blood-brain barrier is only made up of these endothelial cells, but they're surrounded by something called tight junctions. We don't want to have all of this space that would allow pathogens or potential harmful chemicals into the brain, so the astrocytes create this structural support around the endothelial cell and to help tighten the endothelial cells so there isn't any type of leakage that could allow something bad to enter the brain. There's crosstalk between the astrocytes and the endothelial cells. Astrocytes and endothelial cells can also play an important role in producing neurotrophic factors like GDNF. We're going to talk about the importance of neurotrophic factors and how it's related to our behavior and our learning and memory later on in this class. But my main takeaway from this slide that I want you to know is that the blood-brain barrier itself is made up of endothelial cells, and they are supported structurally by the astrocytic end feet. And so the astrocyte end feet help the endothelial cells remain tight so that there are no loosening of these junctions. And this essentially prevents anything bad from entering the brain. So why is the blood-brain? So I've already talked about the importance of the blood-brain barrier because it helps keep blood and other chemicals out of the brain. And this is essential for preventing excitotoxicity, in which cells get overexcited. And we'll talk about what happens during that process. But this can be very detrimental to the brain and cause long-term damage. So there are two different types of strokes that can occur in the brain. You may have heard of them before. One is a hemorrhagic stroke shown here, where a blood vessel can leak and bleed into the brain. This is usually caused from a bursted blood vessel. And so this will allow blood to get into the brain, which is going to cause overexcitation from the certain properties in the blood. An ischemic stroke is different because this is from blockage in the blood vessel that supplies blood to the brain. As you can see, if there's plaque buildup from cholesterol like LDL, this can unfortunately clog the blood vessels and block blood being supplied to the brain. In this case, if you had a loved one that was potentially experiencing a stroke, you would want to get them to the hospital as quickly as possible because there is something called a tissue plasminogen activator, TPA, and this can be used to help unclog or unblock blood vessels. This has to be administered within 3 hours of a stroke. This would not be used for a hemorrhagic stroke, but it could be used for the stroke in which there is clotting from LDL building up. So essentially, if you ever are worried about a grandparent or parent that could be potentially having a stroke, just get them to the hospital as quickly as possible. Because if you could administer the TPA within 3 hours, you could help them and prevent any type of disability, because strokes are sadly one of the key neurological disorders that unfortunately will induce long-term disability. We're also going to just briefly talk about astrocytomas. So this is a tumor formed from astrocytes. There are several different types that I don't really need you to know, but this particular one I wanted to talk about is a glioblastoma multiforme. So this is the most rigorous tumor that can develop in the brain. It spreads incredibly far, and it's very difficult to remove without brain damage. So a glioblastoma multiforme tumor would be the most deadly tumor out of all of them, and this forms from astrocytes unfortunately malfunctioning. So next we're going to talk about myelin-producing cells. If you remember, myelin is super important because it insulates the axons. Axons are those long branches stemming from the soma or cell body, and if they are more heavily insulated by myelin, they can transduce nerve impulses faster, meaning that the neurons can send information to one another in a faster manner. So the first one I want to talk about is oligodendrocytes. And these are a type of glial cell that actually sit outside of axons and help to produce the myelin sheath. Here's an axon, and as you can see, the myelin sheath is very thick in diameter, so this would help increase faster nerve, to speed up nerve impulses along this axon, and this occurs in the central nervous system. So this would be in the brain and the spinal cord. Schwann cells are oligodendrocytes, but they are in the peripheral nervous system. So Schwann cells also help to create these layers of myelin, and in between these segments of myelin are the nodes of Ranvier. So nerve impulses will bounce from each node. And keep in mind that myelin here is going to be generating faster nerve impulses. There are two different autoimmune disorders I'm briefly going to mention here that are associated with malfunction of oligodendrocytes. So multiple sclerosis is sometimes induced by degeneration of oligodendrocytes. And then there is Guillain-Barré syndrome, which can develop after you have the flu or an infection like Epstein-Barr or the Zika virus. And so both of these affect the myelination of axons. And you could imagine how detrimental this could be if you were slowing down your nerve impulses, because it would be preventing the normal communication between nerve cells in either the central nervous system or the peripheral nervous system. And now my favorite cell is the microglial cell. This is my favorite type of glial cell, I should mention, because I know I said that I love Purkinje neurons just because they're beautiful to look at, but microglia, they function as the resident immune cell in the brain. So if you've taken immunology or heard of a macrophage, that's an immune cell that's in your blood and in peripheral organs of the body that can help fight off infection. Similarly, microglia help protect the brain. So you can think of them as active immune defenders of the brain. Microglial cells do function like the brain's resident phagocyte, similar to macrophages in the periphery of the body that are immune cells. So if there's any type of pathogen or debris in the brain, macrophages are in charge of cleaning it up. And in this process called phagocytosis, they will actually eat up cellular debris and ingest it in their system. And so microglia are super protective and important to ensure that any type of pathogen or infection in the brain can be rid of. Microglial cells have other functions as well. They provide structural support to neurons similarly to astrocytes. They can play a really important role in synaptic pruning. We're going to talk more about synaptic pruning as the semester goes on, but essentially, synaptic pruning, you experience that every day. It largely happens when we sleep, actually. Imagine if we had to remember every single thing we learned every day. We form new synapses constantly. The brain is so amazing that it can prune away unwanted synapses so that we don't have to carry too much information with us. And so microglial cells actually play a role in helping with synaptic pruning for neurons. Also, microglial cells, of course, provide immune protection against any unwanted pathogens or bacteria by phagocytosing that debris. This is a very advantageous thing, and it's very adaptive that microglia can protect the brain. In a normal, healthy brain, activated microglial cells are so beneficial. However, in a pathogenic state, they can also harm the brain. It's like a double-edged sword. As I previously mentioned, microglial cells are the immune defenders of the brain, and they can help phagocytose bacteria or debris in the brain. This is extremely adaptive and advantageous. However, if microglial cells are chronically activated, this can lead to a pathogenic state. So here's a great example in the context of Alzheimer's disease. If you remember, we talked about the misfolding of proteins called amyloid beta. Amyloid beta can clump together to form extracellular plaques in the brain and disrupt neuronal communication in the context of Alzheimer's disease. Well, amyloid beta is actually an inflammatory trigger. So microglial cells will register amyloid beta like they would a pathogen. Essentially, microglial cells become activated when amyloid beta is present, and they release neurotoxic factors. This could include something that increases inflammation, known as pro-inflammatory cytokines, or reactive oxygen species. You may have heard of reactive oxygen species before because it promotes oxidative stress in the brain. And there are several different things that can increase oxidative stress in our bodies. Number one is pollution, but smoking and even exposure to UV can increase oxidative stress in the body. So, if there's chronic secretion of these neurotoxic factors, it results in this self-perpetuating neurotoxicity known as microgliosis. So even though microglia are initially advantageous at removing pathogens, if they're chronically activated by Alzheimer's disease pathologies, they'll create more neurotoxic factors that directly hurt neurons and lead to neuronal death. So the big takeaway from this is that microglial activation is advantageous and absolutely essential for the brain. However, in a disease state, microglial cells can further cause harm and disruption to the brain by killing neurons.